Chapter 2, Atoms, Ions, and Molecules, Part 4. So, biological macromolecules can be formed and broken down by various enzymes within our body. If we are synthesizing or making new biomolecules, macromolecules, from their monomers, it goes through a process called dehydration synthesis. So what this means is that when the monomers chemically bond to another monomer, between the two molecules, one subunit will lose a hydrogen, the other one will lose a hydroxyl group, and those two things will form a water. So dehydration, the monomers are dehydrated, they lose a water to form the macromolecule. So the opposite, of course, is breaking it down. So you have a large macromolecule, you want to break it down into smaller pieces. It's involved in a hydrolysis reaction, hydrolysis, breaking water. So as expected, when the two portions of the macromolecule break apart between two monomers, one of them is going to add a hydrogen, the other one's going to add a hydroxyl group. So the water gets broken down between the two subunits. So you want to make more macromolecules, dehydration synthesis. You want to break down a macromolecule, hydrolysis reaction. There are four kinds of biomolecules: lipids, carbohydrates, nucleic acids, proteins. So lipids. Lipids, as we know, are the exception. They do not have monomers. They do not have monomer subunits repeating again and again and again and again. Also, lipids are water insoluble. They are a non-polar type group. So for instance, here we have the classic triglyceride, one glycerol molecule, carbon, 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 and three fatty acids, each attached to one of the carbons of the glycerol. The functions of lipids in our body vary a lot. They can be involved in storing nutrients, storing energy. They can form cell membranes or other cellular components, and they can be hormones. And the main kinds are triglycerides, phospholipids, steroids, and eicosanoids. Triglycerides are the form used for energy storage. It's most common form, most common in the foods we eat, whether it's animal fats or plant oils, and they're stored in adipose tissue. So the main functions, energy storage, but also the fatty tissue can form structural support for organs, can provide cushioning for delicate structures, and insulation to reduce heat loss. Again, you form triglycerides by having one glycerol molecule with its three carbons attached to three fatty acid chains. And the fatty acid chains are made up almost exclusively of carbons and hydrogens, with maybe some double bonds between carbons. The only exception, of course, is the carboxyl group at one end of the fatty acid needed to attach to the glycerol. So you can form triglycerides through dehydration synthesis. One glycerol, three fatty acids come together, form one glycerol molecule and three water molecules, because for each component you needed to lose a water molecule. Fatty acids can vary in length, so some are smaller, some are bigger. They're all pretty long overall. If they lack double bonds, they're called saturated. Saturated because they have all the hydrogens possible to bind to those carbons. If they have one or more double bonds, they can either be unsaturated or polyunsaturated. So example A here, this fatty acid is unsaturated. It has a double bond. B, this fatty acid is saturated. Hydrogens are everywhere possible, again, except for the carboxyl group needed to attach to the glycerol. And then fatty acid C is also unsaturated because it has a double bond. And it turns out saturated fats may not be bad for us anymore. So if you like butter, you can feel safe in eating it. To a point. Phospholipids. Phospholipids are those amazing amphipathic molecules that form cell membranes. They have the charged phosphate head and two long fatty acid chains that are nonpolar. So nonpolar faces nonpolar. So you end up with the phosphobilipid bilayer membrane, 2 layers of phospholipids forming the membrane. So again, 1 charged phosphate portion and 2 fatty acids coming off of it, long nonpolar fatty acids, charged polar phosphate group. Steroids are hydrocarbon rings, multi-ring structures. So here is a generic structure for a steroid. Here is cholesterol. Cholesterol is a steroid. And it turns out most of the other steroid hormones are use cholesterol as a precursor. So cholesterol is the precursor for most steroid hormones. And on top of that, cholesterol is very important in our plasma membranes, the membranes of cells. They will form within the phospholipid bilayer, and they help to add to the rigidity of the membrane. And then finally, eicosanoids. They are interesting, slight ring structure, slight sort of fatty acid chains, not really. They are important for local signaling. So they're very short-lived hormones that can be released to have a localized effect. And this image from the book just shows how you have, say, triglycerides that get broken down in the adipose tissue to release the fatty acids that are carried by proteins throughout the body. So again, you can see how steroids such as cholesterol, phospholipids, and the plasma membrane, and maybe eicosanoids all can affect a cell. Carbohydrates have lots of carbons with oxygens or hydrogens attached to them. So as we can see here, all of the carbons have at least hydrogens and sometimes oxygens attached to them. Their chemical formula is CH2N because, again, for each carbon you have an oxygen, and then you also have 2 hydrogens. The monosaccharides are the simple sugars. So example of monosaccharides includes galactose, fructose, ribose, deoxyribose, and glucose. Glucose being the most important, most common monosaccharide. Disaccharides, on the other hand, are 2 monosaccharides that are covalently chemically bound to each other. Sucrose, simple table sugars made up of a glucose and a fructose. Lactose, found in milk, is made up of glucose and a galactose. And maltose is made up of a glucose and a glucose. Polysaccharides are when you have long chains of monosaccharides. Extremely long, could be hundreds, could be thousands of monosaccharides. So example in plants is starch. Starch is polysaccharide made up of many, many, many glucose monomers. In animals, like us humans, you have glycogen. Glycogen is, again, long chains of glucose molecules, but in this case, they'll also form branching chains off of the main chain. So glycogen is pretty much the main way we store glucose in our cells. Glucose, 6-carbon carbohydrate, 1, 2, 3, 4, 5, 6, forms a lovely ring. And very important to maintain proper amounts in our bloodstream because glucose is the primary nutrient for providing energy to our cells. So our cells need glucose to break it down to form chemical energy that the cells can use to do their various functions. Glucose, again, is stored in the form of glycogen in many cells, liver cells, skeletal muscle cells, and when we need to release glucose in the bloodstream, it goes through glycogenolysis again to release glucose. And when we have tons of glucose, we need to pull it out of the bloodstream and store it. Then the glucose gets turned into glycogen via glycogenesis. So glycogenesis to make glycogen, glycogenolysis to make glucose from glycogen. And again, glucose, say found in the liver, can be broken down and released into the bloodstream and taken to our cells to make energy. And that is it for this part of the lecture.